Okay. I didn't just enter in the meeting. I didn't see how many people were in, and I saw on the schedule, on the program, that the most of presentations are related to infectious disease, but not transmitted by insect vectors. Is that right? So, well, I have only 2 minutes. Let's start. Uh, I'm talking about Trypanosoma cruzi interaction with the insect vector, and I know there will be the people who are participating works with a lot of different subjects. So if anyone have questions, please load it, and we can talk later after my presentation. So for the parasite to succeed on its infection, on its development inside the insect vector, it's not so easy. It depends on a long coevolution. So it depends on the parasite strain and the triatomine species. And this also can be can have other factors such as the immune responses of the insect and the microbiota composition, and also the insect neuroendocrine system. So first of all, I would like to say that I'm going to talk more about the studies related to Rhodmus prolixus. This is a triatomine species that's well studied, and there's more knowledge about Trypanosoma cruzi interaction with this species. The Trypanosoma cruzi, the insects get infected with Trypanosoma cruzi after feeding in an infected mammal, and the Trypanosoma will differentiate in the anterior midgut, is the first one, the number one. It will differentiate and migrate to the posterior midgut. In this organ, it's a very important organ for the parasite infection, because here it differentiates to epimastigote, and this form is a form that replicates. And also, to succeed, the parasite needs to adhere to the epithelial cells. This is also a very essential step for the parasite infection. Later, the parasite will migrate to the rectum and differentiate to tripomastigote form, and it can be released in the feces. So when the insect feeds in another mammal, the parasites in the feces can enter in the little. It can enter where the insect made his feeding when the mammals scratch where it was bited. So, but Trypanosoma cruzi infection is being more important these days by oral contamination, in food contamination. So the insect is important to the transmission, but not only by the feces, the traditional infection from feces on the bite side, but also when the insect contaminates food and the humans drink this contaminated food. So it's the oral transmission. The Trypanosoma cruzi development inside the insect, it only develops inside the digestive tract, and this is very fast. Like in 7 days, we can see most of the parasites here in the anterior midgut or crop. This is the digestive tract of the insect. And here is the image of the parasites, the luminescent parasites inside the digestive tract. So here in 7 days, we see the parasites mostly in the anterior mid-gut, but already migrating to the posterior mid-gut. In 14 days, the parasites are more in the posterior mid-gut, and then with 21 days, they are already in the rectum. They are already ready to be transmitted to another mammal. So this interaction, parasite interaction with the insect, is very dependent on the addition of the parasite to the epithelial cells, the insect epithelial cells. And this can be inhibited by lectin's recognition, and it can depend on the epithelial cell morphology, which is also associated to the insect neuroendocrine system. Okay, so here is to show that the parasite strains can develop differently depending on the triatomine species. So here they are all Hodgkin's prolixus insect, and we infected the insects with 3 different parasite strains. But as you can see here, the parasite DM28C is the parasite that develops well inside the insects. It has more infected insects when we infected with this strain. And the Y strain is not a good strain to develop. And if we do in vitro interaction between the epithelial cells with these parasite strains, we see the same thing, like DM28C is a good parasite strain to adhere to the epithelial cells, while the Y strain is not so good. So that's what's reflecting the success of infection here. Okay. So this addition can also be related to the neuroendocrine system of the insect. When the insect has a blood feeding, the presence of erythrocytes and the distension of the anterior midgut or crop will activate the neurosecretory cells here in the insect brain to produce the hormones, the neuroendocrine hormones, such as juvenile hormone and ecdysone. The ecdysone is very important to the ecdysis. In all stages, ecdysone is here being produced. But ecdysone is also associated to insect reproduction and also to the formation of the epithelial cells in the digestive tract. And when these epithelial cells are well formed, the Trypanosoma cruzi adheres to the cell. But if we give azadiractin to these insects, and this drug inhibits the production of these hormones, neuroendocrine hormones, will stop we won't have a good formation of the epithelial cells. You can see here that these cells are not well formed, and they have some clusters. And here they incubated with Trypanosoma cruzi, and Trypanosoma cruzi wasn't able to adhere to these cells. So when we give this drug to the insect, it inhibits the parasite adhesion and infection through the neuroendocrine system modulation. Here is the picture of how we give the blood to the insects. Here is the insect feeding the blood. We usually give rabbit blood. Okay, so this is an experiment that shows that when we give azadirachtin, here azadirachtin is the experiment symbols, the transparent symbols or empty symbols, and they did this experiment with 3 different triatomines: Rhodmus prolixus, Triatomine fastans, and Diptelogaster maximus, and they gave to the insects 3 different parasite strains: VLE, DM28C, and Y strain. And as you can see here, here is how many parasites infected per insect. That in the insects treated with azadirachtin, the number of parasites infecting is very low. Here, with 10 days after treatment, most of the parasite strains get to 0. But when the insects are not treated with the azadirachtin, the control ones, they have a good infection. So we can also observe this, the effect of ecdysone, the importance of ecdysone to the parasite infection when we take out the head of the insect. So we take the brain where the ecdysone is produced out, and we can see the effect of this of the neuroendocrine system in the parasite infection. So here we have the number of parasites and the number of trypanosomes that are the parasite forms that are effective, and the percentage of these parasites. So when we take out the head, we see non-parasite infection. But when we take out the head and inoculating the insect hemocell ecdysone, so we give the hormone back again, we see the infection like control being reversed. So this adhesion is also related to the recognition of the parasite surface molecules. So in the epithelial cells, there are lectins that recognize the parasite molecules, and this recognition is related to the success of the parasite adhesion. So in these experiments, they incubated different molecules presented in the parasite surface membrane, such as heparin in gray and chondroitin in white, with the blood meal, and they observed that during the days, the insects infected with Trypanosoma cruzi and treated with heparin and chondroitin, they did not have a good infection after 30 days after infection. Only control here have a good number of Trypanosoma in the midgut. So just after feeding, they have an increase in unit-forming colonies of bacteria inside the digestive tract, and around 8 days, it comes to a peak of bacteria growth, and then this bacteria starts to decrease. But when we infect the insects with Trypanosoma cruzi, the bacteria doesn't grow too much. It's lower than comparing to control. We observed that if we give to the insects the parasite strain DM28C, this strain is capable of reducing the CFU in the digestive tract in anterior mid-gut, especially in the anterior mid-gut. But when we give Y strain, this parasite strain doesn't diminish so much the bacteria as the DM28C does. Another thing is that when we treat the insects with a mix of antibiotics, we see a very high number of parasites per insect, per digestive tract, in comparison to control. So when we diminish the microbiota by antibiotic treatment, we see a higher amount of parasites inside the digestive tract. Here you can see by, in our Hodnus prolixus insectary, the main bacteria composition in the microbiota are Serratia marcescins and Rhodococcus hodni. Serratia is, and if we do a PCR, we observe that when we treat the insects with antibiotic, we reduce Serratia marcescins, and when we infect the insects with Trypanosoma cruzi, this parasite is also capable of reducing the amount of Serratia marcescins inside the insect's digestive tract, but not so much as this antibiotic reduced it. In this same, these antibiotics that we gave in this experiment wasn't able to reduce Rhodococcus hodni, but we can see that Trypanosoma cruzi diminished this bacteria species also when comparing to control. So later we studied the Serratia marcescins from our insects from our colony, and we found out two different strains, Serratia marcescins RPA1 and RPH1, and these bacteria, we analyzed their genome, and we observed that they have different types of secretion systems, and they are the proteins presented in these secretion systems are different when comparing RPA1 to RPH1. So maybe these secretion systems are an important factor between the bacteria with the interaction with the parasite. When we incubate this bacteria with the parasite, we see a great increase, a great parasite lysis. It's more intense in the exponential phase of bacteria growth, where we can see that the bacteria Serratia marcescins RPH1 is have a great parasite lysis. Sorry, I told you wrong. In the stationary phase, we see a great parasite lysis, almost in 100% of parasite lysis, especially RPH1 when incubated to Y strain. And when we do a scanning microscopy of this interaction, we can observe the bacteria adhesion into the parasite. Here is the parasite, and you can see this close adhesion. And after 5 minutes, the parasite morphology is already damaged. And so the bacteria has a trypanolytic activity against the parasite, and in the digestive tract, we can observe that the parasite infection is capable of diminishing the amount of bacteria from the microbiota. So another thing is that when we infect the insect with Trypanosoma cruzi DM28C, we see higher amount of antibacterial activity. It's an immune response from insect. And this antibacterial activity is higher against Serratia marcescens, and it's higher when infected insects with the strain DM28C in comparison to Y strain. We also observed the antimicrobial peptides expression in the insects infected with Trypanosoma cruzi DM20C and Trypanosoma cruzi Y strain. And we observed that Trypanosoma cruzi DM increases the expression of defensin B, increases the expression of defensin C, and increases the expression of prolixicin. So the parasite's infection increases the expression of 3 different antimicrobial peptides, and this could be the immune response that is reducing the bacteria in the microbiota. And when we give to insects the parasite with antibiotics, we see, as I already showed, a great amount of parasite in the insect, and defensin C is much highly expressed. Another thing is that the parasite infection also increase phenyloxidase activity, and related to production of nitrite and nitrate that is related to reactive nitrogen species, we see a diminish in this immune response when the insects are infected with Trypanosoma cruzi. Moreover, we analyzed the inhibition of the phosphorylation of NF-κB in the that is presented in the immune signaling pathway of the insects, and we gave IMD 0354 to the insects, and this is an inhibitor of the inhibiting phosphorylation. And we observed that when we give this molecule to the insects, we observe a diminishing in the infection of Trypanosoma cruzi and a higher mortality of the insects, because the infection of these microorganisms increases a lot, especially when we give Staphylococcus aureus to the insect immunodepressed with the drug IMD. Okay, and this drug, it interferes in the amount of bacteria inside the digestive tract of the insect. So we see a higher number of bacteria in comparison to control. And it's here, the dotted line is control insects. So we observed a higher amount of Serratia marcescens and a higher amount of Rhodococcus hodgkini in the insects treated with IMD, in contrast to the group that are only infected with Trypanosoma cruzi. Here you see Trypanosoma cruzi diminishes the amount of Serratia marcescens, and whereas when we give IMD with Trypanosoma cruzi, there's a great amount of Serratia marcescens. So, as we know, Serratia marcescens have trypanolytic activity against this parasite. So, to the success of Trypanosoma cruzi inside the insect vector, this Trypanosoma cruzi should be able to modulate the insect immune responses, especially increasing the expression of antimicrobial peptides and phenyloxidase activity, which will act in the amount of bacteria in the microbiota. So in this way, the parasite is able to complete its infection. How much time do I have? Is everybody listening to me? I don't know when I started, if I can go on. Well, this is just a scan of the parasite infection inside the digestive tract. So, okay, where is it? So here we represent the parasite with commensal and pathogenic bacteria inside the digestive tract lumen. And we know that the parasite can interact with these different microorganisms, and it can be lysed by, for example, bacterial Trypanosoma activity. But the parasite, to complete its development, it needs to adhere to the epithelial cells represented here. And these epithelial cells, this recognition and the adhesion, can induce different immune responses. In some way, we don't know how, the parasite increases some immune responses that can be active to reducing the bacterial load inside the digestive tract. And also, this recognition can activate immune responses in the hemocell, where we find the fat body, that's the important tissue for the immune response, for the insect immune response. And this tissue can stimulate more immune response to go inside to the on the lumen, at the lumen of the insect digestive tract. So I think that's it. I don't know exactly how many time do I have, if I already passed the other slides. Just to explain a little bit more, and that yeah. These antimicrobial peptides, they are especially defensive C. They are strongly related to bacterial inhibition, strongly related to the growth of the bacteria. You see here that the expression was like almost tenfold higher than control. It increased much more than the other antimicrobial peptides. And we speculate that this antimicrobial peptide is essential to regulate the amount of bacteria inside the microbiota. And that's why we see a so higher increase in the amount of bacteria when we, in the insects treated with IMD, because when we inhibit the immune signaling that's associated to the production of antimicrobial peptide, the amount of bacteria increases a lot. And this higher amount of bacteria, especially Serratia marcescens, can diminish the amount of parasites by Trypanosoma activity. We don't know yet how the bacteria lyses the parasite, but we suppose it's related to the secretion systems. And we found in the bacteria genome 4 different types of secretion systems. And in this, you can see that when the bacteria recognize the the other cell, it injects proteins, effector proteins that can kill the parasite, but we don't know exactly if it's this type 6 secretion system that is responsible on parasite killing. Let me see if I forgot to say anything else. Oh no, I took it away. I had one result showing the amount of expression of this secretion system type 6 inside the insects infected. And we observed that when we infect the insects with Trypanosoma cruzi TM28C, we have higher expression of a protein belonging to the type 6 secretion system of Serratia marcescens. So in comparison to control, the amount of expression of this secretion system was higher. So we imagine that the bacteria, when recognize the parasite inside the insect's digestive tract, increase the expression of its type 6 secretion system to lyse the parasite. But to overcome this, the parasite modulates the insect's immune system to eliminate the bacteria. So did I pass 3 minutes or should I talk more? I should have put my time. I can see the chat. Well, here is just an image that I had put, if I had time to show, the molecules presented in the parasite surface, and they are very diverse. And in each of these structures, they have different carbohydrates that can be recognized by the lectins and adhere to other cells. And in these experiments, the authors, they did a parasite, genetic modified parasite, overexpressing in chagazine, which inhibits the parasite adhesion to the epithelial cells. And this is a control of transfection. And this is the white type of parasite. And here, what? And this is the overexpressing chagazine. And so you can see here when we infect the insects with this chagazine overexpression Trypanosoma cruzi, we diminish the amount